Welcome back, everyone. Our second speaker is Dr. David Shapiro, who is a supervisory research entomologist and research leader at the USDA-ARS Southeastern Fruit and Tree Nut Research Station in Byron, Georgia. He is also an adjunct professor at the Department of Entomology at the University of Georgia, as well as the Department of Biology, Fort Valley State University. Dr. Shapiro's research focuses on invertebrate pathology and microbial control. Areas of interest emphasize the study of entomopathogenic nematodes and fungi and include biological control applications, production and formulation technology, and studies on nematode behavior, especially foraging dynamics. David, the floor is yours.
Thank you very much. I appreciate this opportunity to speak to everybody. I'll go ahead and share my presentation. Does that look all right? Everybody can see.
Yes.
All right. So I'm going to be talking about using microbial agents to control insect pests and ways that we're using in my research as well as others to enhance the efficacy of these microbial control agents or biopesticide agents. So first, I want to define what is microbial control. It's the use of pathogens, virus, bacteria, proteus, fungi, nematodes, or their byproducts to suppress insect pest populations. The advantages are these biopesticides are generally safe to humans and other known targets and the environment. Most of them have a narrow host range, though there are exceptions. There's little or no chance for pesticide resistance in many of these biopesticides, and production and formulation technology exists. Some disadvantages is that often the cost of these materials are more than some of the other tactics, particularly chemical insecticides. The narrow specificity can also be a disadvantage if a grower wants to control a variety of pests at once and their biopesticide only kills one species. That can be a disadvantage. Environmental degradation is an issue for many of the microbial agents. Susceptibility to UV and or desiccation will limit the activity of the microbes. So what are the pathogen groups? You have nematodes, fungi, virus, protista, and bacteria. These are the same pathogen groups that attack humans, but fortunately, for the most part, these pathogens or disease-causing agents only attack insects and they don't harm humans and have little or no effect on other non-targets. I highlight the nematodes and fungi because that's really where I'm going to focus today, and especially on the entomopathogenic or insect-killing nematodes, though we have worked in all of these areas, and they're all biopesticide products for most of these. You're probably well aware of bacteria such as the chromobacterium products, Grandival, BT, or Bacillus thuringiensis virus. The nuclear polyhedroviruses are very successful commercially in terms of biopesticide products. But again, I'll be focusing really on the nematodes and fungi. So just give some basic biology on these organisms and then some of the pest targets that have been successful commercially. Then I want to spend the majority of the time really just talking about the research that we do to improve efficacy of these microbial control agents, improving the strain or the organism itself, improving mass production, formulation or application technology, and also trying to gain a better understanding of the biology, basic biology and ecology of the organism. And in this case, the example will be again with the nematodes. And then we can leverage that knowledge to improve pest control down the line with that new knowledge that we have. So the entomopathogenic nematodes, also often called beneficial nematodes or abbreviated EPNs, these should not be confused with the harmful plant parasitic nematodes that can be serious pests or serious pests of various crops. Here in Georgia, we have major plant parasitic nematode issues in various vegetables, soybean, peaches, and so on. These nematodes, the entomopathogenic nematodes, only attack insects. And in fact, they can, to some extent, suppress the plant parasitic nematodes, the good nematodes suppressing the bad nematodes in some cases. For biocontrol purposes, we think of really just two genera, these entomopathogenic nematodes, Steinernema and Heterabditis. They were first discovered in the early 20s. We now have many different nematodes to choose from, and that's important because for any particular insect pest that you're trying to control, there's going to be one or two nematode species that are really better for killing that pest. There's about 20 commercial producers of these nematodes worldwide. And if you see what an insect looks like when it's infected by the nematodes, you'll see these are, for example, waxworms. They nice yellow color when they're healthy. When infected by the nematodes, they change color. So the stunning nematodes will tend to turn them brown or tan, whereas the heterabdidids tend to turn them a reddish color. And the reason for that color change is the pigment in the bacteria that the nematodes carry. I think really probably the most fascinating part of the nematode biology is that they have this symbiosis with bacteria. It's really a partner between the two organisms, the bacteria and the nematode, that work together to kill the host. The heterabditid nematodes are associated with photorhabdous bacteria. Photo because they're luminescent. So you can see this caterpillar infected with the nematodes and bacteria glowing in the dark. If there's time during question answer, I could talk about how we were cited in a segment for a History Channel that has to do with this luminescence phenomenon. The Steinernema nematodes are associated with Xenorhabdous species of bacteria, which are not luminescent. Just a basic life cycle of these nematodes, how they work in nature and when you apply them in the soil. The infective juveniles, which are the only free living stage of the nematodes, enter an insect usually through mouth, anus, or spiracles, the breathing holes, or they can sometimes go through the cuticle of the insect. They then release their partner, their bacteria, and the host will then die, usually within 24 to 40 hours after infection. The nematodes and the bacteria will proliferate inside that dead insect so that you'll get tens of thousands or even hundreds of thousands of new nematodes coming out of that dead insect ready to find another one to attack. The heterabdidids have hermaphrodites, whereas the Steinernema do not. Some beneficial attributes of these nematodes: they kill a wide range of hosts relatively quickly, especially compared to some other pathogen groups like the viruses will take maybe a week before they kill the host. Fungi, maybe 5 days or so. They have this durable infective stage, infective juvenile or IJ stage, that makes them amenable to mass production and application. These nematodes are really safe to humans and other non-targets, so much so that they're exempt from EPA pesticide registration. So if anybody here found some of their own nematodes, and they are all over the place, these entomopathogenic nematodes want to start a company and start selling them. If you produce them, you could do that without any kind of registration, which is not true for the other pathogen groups. So we have a little bit over a dozen commercialized species that have been used in biological control against insect pests. And they're produced in several ways. Most of them are produced in large, this is a small one, but in large fermentation tanks, like brewing beer. And then you can have a solid culture as well, or produce them in insects. So these are dead insects. And in this model that we use in the lab a lot, the nematodes come out and then they go into this water trap where we harvest them. But commercial producers will do that same thing on a large scale. So if you're applying these nematodes, and one thing you always have to remember for many of these microbial control agents or any biocontrol agents, if you're getting a product that's alive, you have to keep that in mind. Unlike chemical insecticides, these organisms can die. And so if you mistreat them, don't handle them in the proper fashion, they'll die and they're not going to be of any help to you. So the nematodes, they can be stored, but generally under refrigeration. Depends on the species, the optimum temperature. A shelf life can be several weeks to a few months, depending on the nematode species and formulation. There's a variety of different formulations that nematodes will come on. Some of the more advanced ones, you really just throw them in your spray tank and ready to go. Others, you'll have to strain them out, the nematodes out, and separate them from the formulation. So you'll have an extra step. Equipment for application, everything from low tech to very high, starting from a watering can to aircraft application. Various sprayers, various irrigation systems, as well as some injection and baits. So there's a variety of ways we can apply the nematodes using standard agricultural equipment. When you're applying them, you'll probably want to take out screens from the sprayers so they don't get shredded. Nozzle size can be important. And then again, with making sure the nematodes stay alive, avoiding excessive heat or settling. They need to move around in the tank, and you don't want them to get oxygen deprived. I just had a peach grower recently who said to me, he applied and he told me after the fact that his tank didn't have any kind of mixing agitation mechanism. So I'm thinking like these nematodes will sink. So you may have mostly been applying water, unfortunately, but any of the normal and most tanks will have some agitation, or you have to do it manually, will keep the nematodes in suspension. Pressure is usually not an issue for the nematodes. They can withstand a lot of it, but it is something to keep in mind. And there are different nozzle sizes that would be better than others, though really, and same with the pumps, really any of them can be used, but some are just a little better than others. So looking at commercial targets, there's some real success stories with the nematodes against some of these commercially important pests, such as citrus weevils, diaprepes, black vine weevil, fungus gnats, mole crickets, and white grubs. These are all excellent targets for the nematodes. And then there are some others like the western flower thrips, chinch bug, cranberry gourd, like codling moth. And there are quite a few others that you'll see on the web. If you look at some company websites, what insects are being recommended for control with the nematodes. What we always come back to is, are you seeing refereed scientific papers that support the field efficacy? We want to see a high level of efficacy in repeated scientific trials in the field. This is just one list of some of the main target insects, and this is abbreviated the different nematodes with all of these where we did see documentation of high levels of efficacy. These are just some that we work on in our lab that we getting very high levels of control. And we work primarily in peach and pecan systems. So the borers, the uh, various weevils, small hive beetle, which is a major bee pest, and lesser peach borer. So just a little bit about the entomopathogenic fungi. That was the first disease of insects found, and interestingly, is the first microbe shown to cause disease, period, when they found the Variabaciana, this white muscadine fungus. It can occur as single cell of branch mycelia. It's grown in fermentation, solid or liquid. It goes right through the cuticle. The spores penetrate through the cuticle of the insect and kill it. And the most important ones are going to be in this group. Hypocreales, you have Bavaria bassiana, the white muscadine. Metarhizium is similar, but a green fungus. And then you also have one called cordyceps. Microsporidia, also a disease of insects that have been used in microbial control, is now actually included with the fungi. I was told that most of the audience is especially interested in turf and nursery pests, so both for fungi and or nematodes. These are some key pests that can be controlled with these organisms. You have a lot of white grubs, the black cutworm, various borers, bill bugs and solid whiteworm, mole crickets, and others. If there's specific questions that you want to try microbial control with, you can certainly email it me. I'll have my email at the end of the presentation. So next, I want to talk about the barriers to expanded use with these microbial control agents and how we try and overcome those. One is the cost, as we mentioned before, and the sensitivity to the environment. There's often a consistency in field performance issue. Well, this happens occasionally where we have to try and make the organism more virulent and more consistent in the field. And then again, we have this basic knowledge we want to use to enhance our efficacy. So our response is direct to those problems. We can improve the organism and its stability, improve mass production to make the organism more efficacious as well as less expensive, improving formulation application, and again, understanding the basic biology. So I'll just give some examples of each work that we've done. So to enhance the organism, you can go through various approaches. The easiest one is just to discover better organisms, just go out and find a better nematode. So we found 2 new species in our lab, as well as numerous other strains, some of which are now commercialized. You could do genetic selection for a particular trait, such as or hybridization, which is crossing the nematodes. So if you think of these two kind of like a breeding program for better nematodes that you can use as a biocontrol agent, that's where this is coming from. We've done a fair bit of that, as well as some of the commercial companies are doing that. You have genetic modification, which has been done with the nematodes, though it's not been commercialized at this point. An example of discovery for the citrus weevil, a major pest of citrus in Florida and and in Texas. This is the commercial strain that has been doing a great job against this weevil pest, but it was the only strain, and we thought we could find better ones. So we went to its original location that was found in Texas and found strains, and this is mortality of the insects. So you can see a number of these are better than the original strain, and a mixture of those was the best one. So now the largest nematode producer is looking at those strains as well. Hybridization, just an example. we had this Italian strain of a nematode that had high virulence against pecan weevil, but it couldn't withstand the heat and desiccation in Georgia. This strain, DD136, had the opposite. So we decided to hybridize it and swap the bacteria to get an improved nematode. So this is mortality of the nematodes under desiccating conditions. You can see that Italian strain does poorly, but all of our hybrids did really well. And similar results with the heat tolerance and virulence remain the same. So now we have this improved nematode. So an example how we get better nematodes through hybridization. One of the, as I say, one of the commercial entities is using that approach. So once you have a better nematode, you may want to move on now to improving production. So for in vivo production, which means growing the nematodes in the insects, the cost is really a major barrier, and so is labor, as well as quality. So to get better quality, we improve the media. So just like us nematodes are with the yeast, so if they have more nutrition in the insects that they attack, then the nematodes will be better. So we can give the insects nutrients that will carry them into the nematodes in tritrophic interaction so that you have better nematodes, better quality, they live longer, they're more virulent. And then as far as the process itself, we look to automate it to reduce labor so that the nematodes, well, the procedures automated everything from producing the insects to the final package. For in vitro or liquid fermentation, the way most nematodes are produced now, you just can improve the media or the bioreactor fermentation parameters, which is something we've also done. Moving on now, so you have better organisms and you have better production systems. The next thing is to improve your formulation and application. So probably the biggest barrier for nematodes is applying them above ground because of their sensitivity to UV radiation and desiccation. Normally, in most cases, you're going to apply these beneficial nematodes to the soil to kill soil pests because soil is their natural habitat. And as long as you keep that soil moist, the nematodes will do well. But if you apply them above ground, again, UV and desiccation could kill them. In the case here, we have this lesser peach tree borer, which is attacking the trees, leaving wounds like this. If we apply the nematodes unprotected without a formulation just in the water, they'll fail to control the pest because they're dying. So we try to look at a bunch of ways to protect the nematodes, different adjuvants that are anti-desiccants, put a little diaper around the wound. That was not necessarily something we're going to recommend, but just proof of concept. Then painting the tree or using a sprayable gel, in this case, a fire gel, which is usually used to protect houses and other structures against wildfire. It stays wet a long time, so we thought if we apply the nematodes in this gel on top, it's going to keep the nematodes moist and protect them from UV and desiccation, which it did. So here you see nematodes with this barricade gel. The diaper did pretty well, but not as well as the barricade. The W is for water only, so these are the formulations without nematodes, and the N is for nematodes. Nematodes alone did poorly, and this is percent live insects, so that here all the insects were killed. So we see the gel plus nematode caused substantial suppression, and we think this kind of thing can be used in a variety of above ground systems against various pests. We saw it would be a hassle to the growers to apply first the nematodes and then the gel on top. You have to apply two things instead of one. But if that gel, as you saw, is really thick, so it wouldn't go through a normal sprayer, So we wondered if we put it down to a lower percentage, this gel, 2%, will it still give protection to the nematodes, but it's be able to go through a sprayer? And that was the case. So this is the nematodes S. carpocapsae or SC with the full rate of barricade or the 2%. You can see chlorpyrifos was the standard against a lot of these borer pests. Of course, that pesticide is going away, but it was the standard at the time. And you can see that the nematodes do just as well as chlorpyrifos without the formulation. Again, the nematodes do poorly. Again, this is percent live insects. You can also use this gel in ground applications if you don't have irrigation. And that was found with the peachy borer, which is a cousin of the lesser peachy borer. This one doesn't attack above ground, it attacks at the base of the tree and girdles down into the root. Major pest. Chlorpyrifos was also the mainstay against this one. Well, we see high levels of control against this insect, but if you don't have irrigation, then you're going to have to do something to overcome that. So we can use that gel here as well. And you see, again, nematodes with the barricade doing as well as nematodes with no irrigation doing poorly. That's percent infestation of the peachy borer. One example of fungi where we're looking at formulations right now with an Israeli team looking at nanoparticle formulation. So the spores themselves, the nanoparticles, will surround each spore and protect it from UV radiation. And that's shown in these graphs against tobacco. Another way to apply the nematodes where we might get greater efficacy is what we call the cadaver approach, the zombie thing with the nematodes. Instead of applying the nematodes in an aqueous suspension, we take the dead insects that are produced and just apply those to the target site where the pests are. And then when the nematodes come out of those dead insects, those are the nematodes that will kill the pest. Interestingly, we saw that when the nematodes come out of these cadavers, the hosts where they're reproducing, they have better survival, dispersal, infectivity, and environmental tolerance compared to nematodes that are applied in water. And that's shown here that you get, this is with percent live insect, which is the citrus weevil, and water applications from a white trap or spray did work, but not as well as the cadaver. Cadavers of the nematodes, this is the dead insect here, they rupture really easily, so we thought of a way to protect them from rupturing on application, which was a clay coating. Other coatings have been developed, such as these plastic pill-like things. Or you could use a hard-bodied insect instead of a soft-bodied one like the mealworms and produce that way. We even made this kind of Dr. Seuss contraption that picks up the insect and drops it onto tape, and then that protects the insects and the nematodes come the side. A novel approach for the entomopathogenic fungi that is really getting a lot of attention, probably going to be more widespread in the future, is an endophyte approach. So these insect-killing fungi, like Bavaria bassiana, this white muscadine fungus, it can control a variety of economically important pests, and it is commercially available. And you apply it also like the nematodes using most just standard agricultural equipment. Well, so for example, in pecan, we have pecan weevil, which you can kill a high level of them by applying the fungi to the ground or to the trunk. However, this fungus can also occur as an endophyte, that means living inside the plant without harming it. So we found that this fungus can live inside corn, beans, tomato, cacao, pine, as well as pecan and there are other crops added to this list. We're the ones that discovered it'll survive in pecan. This is a leaf part from the pecan that was seedling that had the fungus in it, and you could see the fungus starting to grow out on an agar media. So if you have this fungus living inside the plant, it can give protection to that plant against insect attack. And that's what we saw in pecan against some of the pecan aphids, the black pecan aphid and black margin, that the endophyte plants had lower populations of the aphids in greenhouse test. And then some of our studies, as well as others, show that plant disease is also being suppressed from this endophytic fungi, the good fungi suppressing or not allowing the disease fungus to get a hold on that plant. And also increased plant growth has been observed in some crops due to the presence of these endophytic fungi. In terms of another application approach is really just to enhance the pathogens that are already in the system without adding more, or the ones that are already there after application can be enhanced. For example, since the fungi also are susceptible to UV radiation, you can plant clover, and that will give a ground cover and therefore protect the fungus that's going to persist longer. Or you can have crop residue to enhance persistence of the nematodes. So I'm going to move on now to understanding the basic biology of the host, really with kind of one example and how that could benefit in the longer run in terms of leveraging that knowledge to increase the efficacy of the microbial control agent. So we understand the pathogen better, and then we use that knowledge. So one area that we've looked at is how the nematodes find and infect the insect host. You can imagine this tiny nematode. These nematodes, they're just at the border of visibility. If you had a glass of water and they were in there, you'd see little dots in the soil. You would not be able to see them. How does that tiny nematode in what's an ocean of soil find the insect host? How do they know which organism to attack? If you put the nematodes in front of a juicy white grub, they're going to go in. If you put them in front of a centipede, they're not going in, or a spider, they're not going in. How do they know what cues is that insect giving off that's telling the nematode that it's time to attack? And how do the nematodes find that insect? How do they overcome the immune defense? So these are the type of questions that we've been looking at. One of the areas that we've been especially interested in is group behavior and chemical signaling, meaning how the nematodes communicate with each other. So, of course, lots of organisms display group behavior, such as those shown here. There has to be an advantage for the organisms to display group behavior. And for the nematodes, it's clear that this would benefit them because that tiny little nematode up against a giant white grub, it's the David and Goliath type of thing, but you need a lot of them to overcome that immune system of the insect. So we hypothesized they must work in groups, both for infection and for movement, so that they'll stay together for dispersal. So we first looked at infection and found that was indeed the case, that they do infect together. If you have an arena and you put a bunch of insects in a circle and nematodes in the middle, they'll tend to infect just a few of those insects rather than all of them evenly. So they're infecting as a group. For dispersal, we did something similar in a large arena. We put the nematodes in the middle and put the insects around and then sample to see, are they moving as a group or are they just moving randomly? And so we did see aggregative of group movement in all the nematode species we tested. So similar to the infection behavior, we saw that the nematodes not only infect together, they move together. So we like to think of them now as these little packs of nematodes in the soil, like a pack of wolves. And we think that we know that there's chemical signaling going on that's driving their movement. We think maybe there's a little alpha nematode leading the pack. But these chemical signals are pheromones. And the next step is like, how do we leverage that knowledge to improve biological control using these pheromones? So the first discovery was then just discovering what are the pheromones and the chemical nature of these pheromones. The scaricides was determined and then shown. So if you have the nematodes and you put the pheromone on an agar dish, you see them disperse. So you see that's active. But that was only in a tiny arena. We didn't know if that would really work in a soil profile. So we wanted to see if that's going to translate into improved insect mortality. And so it did. We see that the percentage larval survival of pecan weevil, this is the control, so almost 100% survival. And then this is nematodes by themselves, which did reduce relative to the control. But then if you have the nematodes plus the pheromone, you get even further reduction. So the nematodes are helping, the pheromones are helping the nematodes move through the soil and enhancing the infection, so you get better efficacy. So there's an example of how we started with kind of basic studies, and then we're leveraging that knowledge to improve biocontrol. In conclusion, we see that microbial control agents do control a wide array of insect pests. Efficacy can be enhanced by improving the organism's production and application formulation systems, as well as basic research. Some of the research we'll be doing in the future is, can we advance automated production further? Is it feasible to have better in vivo production systems? Can we make some systems that the farmers can use themselves to produce their own nematodes? Further development of protective formulations and application methods. For the gel formulation, the barricade that we use is very effective. It's a little bit expensive, so it's going to add to that cost for the grower. So we want to find less expensive formulations, and also ones that will be compatible with organic crops. And then also, how can we further advance infection behavior to improve biological control? We'll also be looking further at the endophyte approach. To what extent will this long-term suppress disease in insects if it's in a tree or in a plant? So far, we've seen maybe 3 years at the most maintaining of that fungus inside a pecan tree. So those are some of the things that we'll be looking at in upcoming research. I want to acknowledge technical assistance, especially my lab manager, Stacy Bird, who's who's sitting in on this webinar right now. All of co-investigators from various institutions, especially University of Georgia, University of Florida, and Brigham Young University, various funding sources, as well as professors Joseph and Kunisi that invited me, and Rich Raymond for helping with the setup of the webinar and all of that. And that's all I have. I'll be happy to answer any questions. Thank you.
Thank you, David. This was wonderful and lots of fascinating information. I was like, wow, this is a fire hydrant. I'm trying to drink a little bit. Yes, encourage everyone to think of good questions and ask. And I have several myself, so I'll just give you an opportunity first to come up with something before I jump in with mine.
And I did have one more slide that had my email that I didn't put. So it's just really simply David Shapiro at USDA.gov. So if anybody has questions after this, feel free to get in touch with me.
I guess the first question I had was you mentioned that you could share with us the image of that symbiotic bacteria bioluminescence, which sounded really fascinating. So if you can show something like that to start us off thinking, that'll be great.
That was just an anecdote I was going to add
Okay.
about the connection with the History Channel and this bioluminescence thing. So try and be brief on it. But so during the Civil War, there was a Battle of Shiloh where the very bad battle, and there were many wounded soldiers, and some soldiers were said to have glow in the dark wounds, and that of course freaked the soldiers out. But they noticed, or reportedly anyway, that soldiers that had these glow in the dark wounds survived better than those that didn't. So then there was a hypothesis that luminescent bacteria, Photorhabdus, which does produce antibiotics, was responsible for that. So the History Channel was doing a little segment on that. And this was, I think, near the beginning of 2020 when they started on it. They contacted me and they wanted to come here to film because they said, can you recreate the glow in the dark insects and bacteria? And then I was like, yeah, sure, we do that. So they were on the come here, but then because of COVID, they were in New Jersey. They can come here. They didn't want to travel that far. So they said, do you know anybody else in New Jersey that might be able to help us? I have a close colleague at Rutgers University in New Jersey who does this kind of work. So I'm thinking like, do I know anybody or do I make them come down here? But I did the right thing and told her about my friend, Dr. Kopenhofer, and so they did go to his lab to film it, but we supplied the bacteria. So it was our bacteria that was on the TV. And we did get an acknowledgement at the end of the segment. So that was nice. So that's the story on the luminescent bacteria.
Fascinating. So that was truly a Georgia grown bacteria you provided there, right?
It was. Yeah.
You mentioned, I guess this is just something that I'm really curious about the endophytic fungi. And I remember from years ago my own studies, Bavaria bassiana, but I didn't realize that it actually can grow inside the plant tissue without becoming a problem. That's really fascinating. And you mentioned that you had looked at aphids. Um, have you done work or know of other scientists who have looked at other pests and perhaps diseases in addition to insect pests?
So we saw Phytophthora being suppressed in our trial. We're repeating that soon. A number of plant pathogenic fungi have been suppressed, like in cotton, different aphid species, some weevil species have been suppressed with the endophyte approach. It's worth testing in each crop, and it could vary. We see that not only that white muscadine fungus Bavaria can be an endophyte, so can Metarhizium, the green muscadine fungus. It's a matter of screening which one is the best, but there is interest in among the pecan growers now to see if that could be used to protect their crops.
Sure. And is the fungus, where is it located in the plant? Is it like in the epidermis, on the leaves, or is it deeper in some tissues? Is it in the
Yeah,
roots?
we found it in the stem, leaves, and roots, and we think it's being carried systemically in the tree because if we started in the soil and the roots, we're finding it up in the leaves too. And interestingly, I was a grad student when this was first discovered in my major professor's lab at Iowa State. He's the first one to report endophytic Bavaria fungus, the insect killing fungus. But I was focused then on my nematodes, and they were really busy with this amazing new discovery. And I like just had my blinders. So then it was funny that many years later I came back, well, that's pretty cool. I think maybe I'll work on that too.
It all comes around, right?
Yeah.
So is the fungus, I'm just trying to imagine, just visualizing within the plant tissue itself. Is it in the cells, in the living cells, or is it in the intercellular spaces, or is it in both? Is it in like dead xylem tissue versus living phloem tissue? Do you know that? I'm sure you do.
I'm sure I don't. They're really good electron micrograph pictures of the fungus. That was one of the things they did first, really verify, but I don't remember where to the cell that fungi were located, whether that was in between the cells is what would be my guess, because it seems like it'd be disruptive otherwise. But I would have to go back and look at those papers to see that.
Yeah, I may have to do that myself. You just got me all interested in how this happens and how
Yeah.
widespread it is in the plant. It just anything that is in the plant tissue, the plant is going to figure out, anthropomorphically speaking, a way to cope with it. But it certainly seems like there could be an active way to utilize this resource and have it in places where, say, an attack organism has occurred, whether it's a disease or an insect, and then have it more there, like the medicine is right there where it's needed. So that's, but then you said it's systemic, so that would make sense that it's everywhere. But yeah, that's really fascinating. And you said there are three, so Bavaria bassiana, and there were a couple of others you mentioned.
Metarhizium is another genus that's been used against insects a lot, and we found that to be endophytic as well as you can spray it. Right now, Bavaria is probably the most common commercial product you'll find. Metarhizium, in fact, I think is pretty hard to get in the US. And then there's another one, cordyceps, which used to be called Isaria. It was called Bacillomyces before that. They keep changing
They
the
do.
fungal taxonomy. But they're all commercial products of the cordyceps. And they have been found to be endophytic to a lesser extent. But they're used like Isaria is especially used for salt-bodied insects like whiteflies. And if Bavaria will do okay against those two, but probably more used for the beetles and the pillow pests.
And which stage of the insect does it attack? All stages, perhaps less eggs?
You mean the fungus?
Yes.
Yeah, the fungus, other than the egg, it can attack, depending on the insect host, it can attack various stages, but it may be more virulent to one stage than another. And it's really the same with the nematodes. We did find that the nematodes, at least in one caterpillar case, will infect the eggs. Um, but more in it, but in many cases, it's going to be the larvae or the adult. But it does really vary. Like in pecan weevil, it's the adult insect that's the most susceptible. But in some other insects, it's going to be the opposite. And then it's a matter of where the insect is. If the adult is above ground, but it's overwintering in the soil, you'll probably do better applying to the soil and killing the insect there.
So there's so many things to keep in mind to maximize the efficacy of a treatment and an application.
Right.
Any questions from the audience? Okay, I'll go with another one. So how do you hybridize a nematode?
We do it on a sterile agar dish. You have to have their bacteria in order to for them to live. But you want to be able to see what you're doing and keep them alive. So you'll have a lawn of their bacteria, and then you'll take maybe 5 or 6 males and a female and put them on the dish, and then wait for the progeny to come, and then you just screen those progeny for the beneficial traits that you're after.
So let me understand this. Those are two different species of nematodes?
No, that's going to be us. Yeah, that I should clarify is only you can different strains. So you'll have one strain of
The
this
same species.
because there's
Gotcha. I understand.
yeah, yeah, there's a lot of variation in capability within species. So you might have a strain that's especially virulent, especially environmentally tolerant or high yielding. And then you try and, just like I said, that if you think of a breeding program like we breed corn or cows or whatever, trying to get to something similar for the nematodes.
I understand. So lots of little things that you can't even see. I mean, they're not visible to the naked eye.
The infected juveniles would be, they're like half a millimeter. So if you have a good sight in a glass of water, you'd see the dots. The adults are about twice as big, so you'll see those like on an agribush. Uh, but you may see that infected with juveniles too, depending on the contrast. But
Very small.
yeah,
Any questions? Any other questions? Seems like I just had a really long conversation there at the end. Um, I'm sure the audience had some questions. Maybe they'll follow up. So do you mind putting your email in the chat so that we can have it? Just type it there.
there you go. And yeah, any questions on the use of the entomopathogenic nematodes or fungi against whatever target pests you're dealing with, we should be able to answer that.
Great. Thank you so much. That was really wonderful information. And I want to thank both Jason and David for joining us today and get us started off to a great 2022 year and getting the best of pests webinar. Thank you again. Thank you all for joining us. And don't forget to chime in again a month and a half from now. Thank you.
Bye bye, everybody.